Thank you all for being here today. And I'm going to tell you a little bit about my research studying the neuronal cell type susceptibility in Alzheimer's disease. So as a neuroanatomist, my research is focused on understanding the structure of the brain and how it's disrupted by neurological diseases like Alzheimer's. The foundation of the brain's architecture is the neuron, a cell which is specialized for coordinating and communicating everything you have ever experienced in your lifetime. To process all of that information, the human brain contains a densely interconnected network of 86 billion neurons, of which neuroscientists like me have struggled to define all of the different types that exist. On the right side is an example of a single neuron with its tree-like branching dendrites arising from a single brightly lit cell body. Along these branches, you can see tiny spines jutting out along each entire length, which each of these spines receives signals from another neuron somewhere else in the brain. And so you can see how a single neuron is processing and receiving information from thousands of others across the entire brain. In neurodegenerative diseases like Alzheimer's disease, these spines begin to retract, the neuron branches begin to shrivel, and ultimately the neurons will die, leaving a silent disconnection in that brain's network. One of the brain regions which is most affected by Alzheimer's disease is the hippocampus, which is critical for learning and memory. The hippocampus is composed of multiple subregions located deep within the brain's medial temporal lobe. And its name is derived from its really unique structure, which is shaped like a seahorse. Within the hippocampus of Alzheimer's patients, neurodegeneration is believed to be caused by the toxic accumulation of amyloid and tau proteins. Amyloid proteins aggregate to form large plaques in the spaces between neurons and disrupt the neuron's local environment. Neuropathologists have described multiple types of amyloid plaques based on their appearance, and still new types of plaques are being discovered and reported every year. In contrast, the tau protein tend to aggregate as threads in the extracellular space, but also tangle within the neurons intracellularly, which clogs up the cell's ability to transport molecules along its dendrites. However, amyloid and tau pathology don't suddenly appear everywhere in the brain. They slowly spread from brain region to brain region through these neural connectivity pathways over the course of the disease. So here you can see an example of hippocampal tissue sections from 3 ADRC brain research study patients that were diagnosed with low, intermediate, and high degrees of Alzheimer's disease. The varying distribution of amyloid pathology suggests that some brain areas and their neurons are particularly vulnerable to the amyloid toxicity, while other areas are more resilient. So the goal of my research is to really map and characterize the distribution of different types of amyloid and tau pathology across different parts of the hippocampus and identify regions with neurons which are susceptible to the disease. So as part of the pilot project with the USC ADRC, our lab is digitizing and archiving the ADRC patient tissue sections and developing new machine learning algorithms to rapidly quantify and characterize the Alzheimer's disease pathology within the hippocampus. So if we zoom in on this area of the hippocampus, we can see that there's 3 different types of this red amyloid labeling in the brain. You can see examples of large, round, dense plaques located here, areas with more diffuse red coloring, and finally small, if you look closely, small red labeling within individual neurons. So as much as we can see this with our own eyes, we can also teach a computer algorithm to recognize these patterns and automatically classify and quantify their features within the tissue image. So in the example on the right, this image has now shown that all of the red coloring has been colored yellow, pink, or purple based on the localization of a dense plaque, diffuse plaques, or the upregulation of intracellular amyloid. So to map this pathology within specific hippocampal subregions, we also used a software program developed by our collaborators in the USC Stevens Neuroimaging and Informatics Institute called the Quantitative Imaging Toolkit to manually draw the anatomical boundaries of the hippocampus subregions. Ultimately, this segmentation mask allows us to localize the different types of pathology identified by the machine learning algorithm into these color-coded hippocampal subregions and provide a quantification of the pathology that was not previously possible. So what do we find? So the amount of new data that we can generate from this process is beyond what I can discuss within this time period, but I'll try to briefly summarize the major findings that we're seeing. So as expected, Alzheimer's disease pathology was higher in almost all hippocampal subregions in patients diagnosed with high AD compared to low AD. However, when we look at the type of pathology that's accumulated across the course of the disease from low, intermediate, and high patients, the diffuse amyloid plaques appear to more rapidly accumulate across the course of the disease compared to the tau tangles, the intracellular amyloid, and the dense amyloid plaques, which suggests that much of the amyloid burden that's being produced in the brain across the course of the disease is due to the production of diffuse amyloid plaques. So one of the greatest risk factors for Alzheimer's disease is the APOE4 allele. And many studies have characterized the fact that this allele leads to earlier onset of Alzheimer's and a greater risk of contracting the disease. So we wanted to look at the patient population to examine patients which carried this APOE4 allele versus patients that did not carry the APOE4 allele. And what we found is that when we quantify all of this pathology, the dense amyloid pathology and the diffuse amyloid pathology, and also looked at the high cases versus the low and intermediate cases, many of the subregions in the low and intermediate cases of Alzheimer's disease show elevated levels of amyloid in the APOE4 patients, suggesting that this corresponds with the higher onset of Alzheimer's disease that these patients experience, or the earlier onset of the disease. Many of these areas, for instance, the subiculum, the prosubiculum, the medial entorhinal cortex, suggest that these neurons are particularly vulnerable to the effects of APOE4 and the production of amyloid. So another area we wanted to look at was that Alzheimer's disease is known to disproportionately affect women versus men. And nearly 2/3 of all Alzheimer's patients are women. So however, despite this increased risk of Alzheimer's disease, we found very few differences in the presentation of the amyloid pathology within the hippocampal subregions, suggesting that there might be other brain areas which are involved in mediating the sex differences in AD. So finally, I want to leave you with a little bit of a future direction that we're working on. So in addition to working to correlate this data with the MRI imaging that you heard about earlier, we're also trying to investigate another brain cell that plays a critical role in Alzheimer's disease and supporting healthy neuron function. These are the microglia. The microglia, which you can see here in brown, which have a very important role in supporting healthy function. So recent studies have shown that the microglia play a major role in the formation of the dense amyloid plaques. And animal studies have shown that if you remove these microglia from animals, that no dense plaques will form and only diffuse amyloid labeling is present. So what we want to do now is look at the microglia distribution and determine if the areas where we see dense amyloid is more correlated with higher microglia density, and areas with the diffuse amyloid is more correlated with areas without microglia. So it suggests that the presence of diffuse and dense amyloid are related to the presence of microglia in the tissue. So in conclusion, I showed you how the digital pathology approaches and machine learning approaches that we're developing within the ADRC allow for this rapid characterization of AD pathology across hippocampal subregions, and that provides new insights into the progression of AD across the brain. The diffuse amyloid pathology we found increased more rapidly across the AD stages compared to dense amyloid or the tau neurofibrillary tangles in the human hippocampus. We found that the APOE4 genotype was associated with significantly increased dense and diffuse amyloid in some of the low and intermediate Alzheimer's patients, suggesting that there are very specific cell types which are susceptible to the pathology in these areas. And finally, we found very minimal differences between males and females as far as the presentation of the amyloid pathology. So I just want to thank you. Thank you all from my entire lab. I really appreciate your participation in this study, without which none of this is possible. So with that, I'm happy to take any questions.